Okay. Hi everyone. Thank you for having me. My name's Jillian McMahon. I am a third-year bioengineering PhD student, and it is my second year on the Interfaces training grant. So it's been a lot of fun. I am co-mentored by Joe Schoenberg and Romy Amaro for this training grant. So today I'm going to talk about the work I've been doing over the past few years, looking at how the mitochondrial network dynamics change through the development of human lung and intestines. So the process from stem cell to specialized cell, let me see if you can see my mouse there. Stem cell to specialized cell is called differentiation. And in differentiation, people typically think of two things, and this is gene expression and extracellular environment. So what genes are being turned on or off, and whether outside the cell is soft and squishy or hard. But there's been recent research showing that the mitochondria actually play an instrumental role in this differentiation pathway. So if the mitochondria cannot restructure, does not look right, the cell cannot continue along that differentiation pathway. And this does make sense because when we look at all the different cell types, they have different mitochondrial morphologies. So whether we're looking at fibroblasts or ovaries, myoblasts, myotube, neurons, they all have mitochondria that look pretty different compared to each other. Also, if we have a single cell type under different drug conditions, the mitochondria again look different. So this leads us to think that different mitochondrial phenotypes actually encode information about what's going on inside the cell and reflect the cell's health. So we know the mitochondria produce about 95% of our energy, in addition to being involved in a variety of cellular processes like apoptosis, immune regulation, calcium homeostasis, all of these things. The mitochondria are very involved in all different aspects, or in many different aspects of cellular function. But this bean-shaped thing is not really what mitochondria look like. And it's misleading because when you Google mitochondria, all you see are the beans. But in reality, they're 4D networks. So when we image in 4D, meaning X, Y, Z, and time, we see that the mitochondria form this three-dimensional network around the cell. This gap right here is the nucleus, and it's very dynamic. So we see it moving across the nucleus with this little piece here. It's undergoing fusion and fission, and it's very dynamic over time. So in order to study a dynamic network like this, we need to use 4D cell biology. Typically for 4D cell biology, people are using confocal microscopy. So with confocal, this one is showing a point scanning confocal. We're illuminating a single point in the organoid with laser, and then that's exciting our single point, fluorescing coming out of the tissue. So if we want to collect, say, a volume that's this size, it's going to take a very long time because we're going point by point by point by point. And then by the time we've gone to the end, the beginning is kind of different. So conversely, our lab uses lattice light sheet microscopy. So this sends in a sheet of light and then detects orthogonally to that sheet, which allows us to be less damaging to the tissue because we're not exposing parts of the tissue that are not actively being imaged. And it allows us to move a lot faster and gives us much better temporal resolution. So we can image our tissue for a lot longer with much better temporal resolution. Our setup also has adaptive optics, which allows us to correct for the aberration that occurs when we're imaging tissue a lot of the time. So this essentially, we know how the wavefront is distorted, so then we can correct for that distorted wavefront using a deformable mirror. And with this technology in hand, let's go back to my original question, looking at the differentiation pathway and the role of mitochondria in this differentiation. So I wanted to know if we can actually predict what the cell fate is, predict what's going on inside the cell in relation to development based on the mitochondria. So I grew lung organoids and intestine organoids from stem cells, which looks like this. And in more detail, I'm starting with stem cells that have been CRISPR tagged with GFP on the mitochondria. and RFP on the cell membrane, which allows us to visualize each of those things with our microscope. The lung and intestine both go through the definitive endoderm pathway, and they look pretty similar here. This thick monolayer of cells releases little buds, and for the lung, this is the anterior foregut endoderm. For the intestine, mid-hindgut endoderm. These buds further differentiate into mature lung organoids and mature intestine organoids. And what's kind of interesting about the intestines is that they're largely made up of intestinal stem cells. So then they continue to proliferate. So these intestines actually, they get bigger and bigger, and then we need to passage them every 7 to 10 days. So that's everyone else in the lab is excited or is jealous of whoever is growing the intestines because we don't need to start at the beginning. So I went through and imaged with our lattice, and this is what that looks like. The green is the mitochondria, the red is the cell membrane. We're going through the Z slices here. We stack up the Z slices to get our full three-dimensional volume. Then we can image over that three-dimensional volume over and over again to get our movie. So this is about a 2-month-old lung organoid. We're looking at about 240 cells here. And we can actually see the dynamic mitochondrial network when we're imaging in 4D. Since we collect the cell membrane data, we can actually segment individual cells within our living tissue and then get subcellular data of the mitochondrial network inside single cells inside of a living tissue with cells in their more native environment. So this allows us to work with data that hasn't really been worked with before. So it's been really cool, really exciting. Every time we see new data in the lab, everyone gets really excited. So I went through and I imaged for each of our developmental time points, and here's some examples, some representative data from these time points. So we have our stem cells, we have our definitive endoderm, we have our young to more mature lung organoids, young to more mature intestinal organoids, although more mature can be argued because it's continually proliferating. So, and I mentioned that we want to extract single cells out of these organoids. So to do that, we developed a new pipeline where we start with our lattice data here. So here's top and side views. And then we use CellPose to go through each Z-stack and segment each of the cells. And then we also do that from 0 to 60 because that's our full movie is 60 time points. And each segmentation is done independently. So Z-stack 20 is segmented independently from Z-stack 21. So we need some way to correlate the labels. So we got our cell centers and used dbScan to cluster them through space. And then we can create our three-dimensional point clouds from this, which exists for each time point. After that, we need to connect the labels over time. So we do frame-to-frame tracking here. Then we can generate these 4D single cell masks for every cell in all of our organoid data. And here's some representative cells from these developmental time points. So we can see already that there are some differences between the lungs and the intestines for the different developmental time points. But this isn't really good enough to convince people. We can look at all these pretty pictures, look at all these movies, all we want, but it doesn't really mean anything until we can put numbers to it. So our lab also developed something called MitoTNT, mitochondrial temporal network tracking, which was developed by Zach, who is right there. So with MitoTNT, we start with lattice data. Again, we can segment an individual cell, and we have mitochondrial data for each time point. So this is our 3D time lapse. Then for each time point in our data, we can segment the mitochondria. From this mitochondrial segmentation, get our skeleton. So these skeletons across time are what actually get tracked with MitoTNT. Our blue is our current frame and our red is our next frame. Black are the tracking arrows. So this allows us to put numbers to all of the beautiful videos that we see in our data. So MitoTNT quantifies on the fragment, branch, and node level. So a fragment is a full network that looks like this. A branch is basically an end-to-end right here. And a node is equally spaced points along the fragment or the branch. So mito-TNT gives us values for the lengths for the fragments and the branches and diffusivity on all three levels and looks at fusion and fission. So since we're studying development, we wanted to look at what people believe is happening with the mitochondria as cells differentiate. And the literature says that stem cells have fragmented mitochondria and differentiated cells have elongated mitochondria. So we were a little surprised when we saw the opposite. Our data shows a sign flip in this trend. So here we have our branch to fragment ratios. If we have a branch length over a fragment length that is equal to 1, then that is not networked because the branch length is equal to the fragment length. If we have a branch over fragment that is less than 1, then we are networked because our branches are shorter than our fragments. So the trend that I saw here, in black, we have our stem cells. This is the most networked of our cells. These cells have the highest level of networking compared to the lung and the intestine. The lung, as we mature in lung cells, we get darker blue. As we mature in intestine cells, we get darker pink. And the lung cells are the closest to approaching 1. The intestine kind of lie between the lung and the stem cells. So we see this reflected in the data too. If we go back and look at our cells, the stem cells have higher levels of networking that's visible. Our intestines have some level of networking, but our lungs have basically no networking that we can see. They're just lines of mitochondria along the side of the membrane. So our belief for why people have seen the opposite is that so much of biology has existed just looking at 2D Z slices, oftentimes in monolayer cells. But we're looking at organoids, which are showing cells in more of their native environment, and we're looking in 3D and 4D. But when we look in a 2D Z slice, it's very easy to say, yeah, all the mitochondria are punctate. All we see are these little dots in the cell. But then when we expand to our 3D volume, we can actually see these networks forming, these three-dimensional networks forming in each of the cells. So we're not just adding the third spatial dimension, we're also adding time. And when we add time, we also see some trends through development. So first, we look at fragment length versus fragment MSD. MSD is just how much something moves in a given amount of time. And we see that larger fragments are moving less and smaller fragments are moving more. So this makes sense because if you have something big, especially in a place as crowded as the inside of a cell, it's going to be harder to move it across the cell. And if we have something small, it's going to be a lot easier to move it across the cell. So this relationship, I thought made a lot of sense just from a physical background. But then if we also consider the like the color of the data, so the blue for the lung, pink for the intestine, black for the stem cell, we see a trend of stem cell into intestine into lung, where our stem cell data shows very little movement in the mitochondria with high levels of networking, and our lungs show much more movement of the mitochondria with less networking, and our intestines lying in between. And if you remember in the beginning, we know that the intestinal organoids are largely made up of intestinal stem cells, and these are going to proliferate a lot more than the lung cells are, because we do need to passage them every 7 to 10 days. So these cells are going through mitosis much more than these lung cells are, which is my belief for why they lie here in between the lung and the intestine, or the lung and the stem cell. So just to recap, as the cells get older, we have lower levels of networking and higher fragment diffusivity. We can also, since we're segmenting individual cells, quantify in relation to the single cell. So look at where the mitochondria are in relation to the rest of the cell, essentially. And what we see is that the stem cells are short and wide, and the lung cells are tall and narrow. So in this shift, the cells are becoming smaller and more columnar, and the mitochondria are moving closer to the cell membrane. So this is another sort of quantification that we can do, is looking at where the mitochondria lie in relation to each other. So we see these trends comparing lung and intestine, and we see these trends through development. So then we wanted to see if we can actually predict something about the cell's information, about the cell's identification, and really link the mitochondria to a cell's identity. So we worked with random forest models, and first we wanted to see if we can predict a cell's type, so lung or intestine, and a cell's age from the lung from 0 to 130, based only on morphology. So this is looking at fragment length, branch length, things like that. And we got pretty good accuracies, I think, about 70%, 80%. And then we did it just with the temporal. And again, we get a little better accuracies. But we found when we combine both the morphological and the temporal features, we can actually get the best accuracy, and with 83.6.7%, predict actually what cell type the mitochondria belongs to and how old that cell is, showing that the mitochondrial network dynamics and morphology are actually linked to what's going on inside the cell and potentially a cell's function. So the main results from this project, I think they have two sides. On the biology side, we showed that the mitochondria become less networked, more motile as the cells specialize, which is opposite of what the field believes currently, showing the need for new technologies and really going back and characterizing what's happening in cell biology. And we see that the mitochondrial network encodes information about cellular health and function, shown through our ability to predict a cell type and a cell age based on the mitochondria. So on the method side, we showed that if we use organoids with this lattice light sheet technology, we actually get interpretable results. It's worth doing. We've established the methods to make this doable for other people as well. And yeah, we have a new method for analyzing live organoids at a single cell scale. A lot of organoid research is endpoint analysis, where people grow the organoid to a specific time point, they kill the organoid, and then they do their analysis. This allows us to actually look at living organoids, look at living cells in their native environment. Yeah, so this research is part of our recent paper. It's available on bioRxiv, and there's a QR code for it if you want to look. But none of this would be possible without this support from my lab, the Schoenberg lab. So thank you all so much, and thank you for listening.
I would love for y'all to have the time to ask Jillian questions, but we've got another presenter that we have to try to fit in here, so we're gonna have to just follow up with Jillian on an individual basis. Appreciate you coming. Thank you so much.
Hey everybody, I'm Catherine Hatch. I work with Dr. Samara Reck-Peterson and Elizabeth Villa on my project. Oh, and I'm a third year in biomedical sciences. And my project the past couple years has been working on understanding how this protein LRRK2 is activated and becomes hyperactive in Parkinson's disease using this new tool called DARPins. Before I really get into that, I just want to highlight that this has been a really big collaborative project and highlight some other names. Andreas Lechsteiner here at UCSD, Stefan Knapp, and Andreas Pluckthun, who have been really instrumental also in getting all of this data or not getting this data, but in allowing this project to happen and giving me a lot of support. And I'll make sure I, credit people where due. But so to get started, Parkinson's disease is actually the second most prevalent neurodegenerative disease worldwide, right after Alzheimer's. It affects around 11 million people worldwide. Last time there was kind of a survey, the most recent numbers. And it's hallmarked by both cognitive impairments, such as dementia, having sleep disorders, sensory issues, as well as kind of those well-known physical infirmities such as that slow movement and that kind of hallmark shuffling gait that everyone thinks of when they think of Parkinson's disease, as well as kind of like a loss of balance and issues walking, as well as tremors. And it's of course hallmarked and was first recognized due to that loss of dopaminergic neurons in the substantia nigra. And although 90% of Parkinson's disease cases, there's actually no underlying cause. They don't know why Parkinson's disease cropped up. About 10% of cases are hereditary, so they come from someone in your family. And of those heritable versions of Parkinson's disease, one of the most commonly mutated genes is LRRK2, this leucine-rich repeat kinase 2. And it's a really large protein. It's got a lot of different domains, starting from the C-terminus with this WD40 protein-protein interaction domain, where it attaches to a kinase domain, this core of ROCK domain, and then a ROCK GTPase. So it is rather interesting in that it's got two catalytic functions. It both has a kinase and it has a GTPase, which converts GTP to GDP. And so I'll be referring to this catalytic half of LRRK2 as RCKW sometimes down the line in the presentation. And so this kinase, it phosphorylates a bunch of different substrate, but really just RABs, but it can phosphorylate RAB5 and RAB3. RAB3 specifically is very common in the brain. It's probably the most prevalent substrate in the brain because it's part of secretory vessels. But it can also phosphorylate RABs that are on membranes in the Golgi apparatus, on the endoplasmic reticulum, and early endosomes. And most of the mutations in Parkinson's disease most of the mutations in LRRK2 that lead to Parkinson's disease are in this catalytic half of LRRK2, which makes sense if you think about it. It's the kinase that's becoming hyperactive. So it's the catalytic part of LRRK2 that is mutated to cause that hyperactivity. But interestingly, I mentioned that there are these genetic or these heritable versions of LRRK2 that lead to Parkinson's disease, but LRRK2 can also be hyperactive and hyperphosphorylate RABs in sporadic forms of Parkinson's disease, so where it's not a heritable form. And so it really makes it, both that fact and the fact that it's a kinase, which are fantastic drug targets, makes it a really nice, important target for trying to treat Parkinson's disease. But as I'm sure you noticed, there's still more to this protein. Again, it's a really big protein. There's the LRR region, which is what LRRK2 is actually named after, the leucine-rich repeat domain, this ankyrin domain, and this armadillo domain. And now, interestingly, based on structural data from cryo-EM studies, when that arm is draped across the kinase domain, I clicked that too fast, but RAB substrate is actually not able to access the kinase active site because it is blocked from where that arm is draping across the kinase domain and the kinase active site. So this really flexible arm has to be kind of undocked from where it rests across the kinase domain and open up for RABs to come in and become phosphorylated. And this arm is really flexible. It's not necessarily just in one spot most of the time. In fact, that RCKW part of LRRK2, that structure was solved before the full-length structure because that arm is so flexible. So in cryo-EM, that arm is everywhere, and so it kind of averages out when you're trying to solve the structure with cryo-EM. And so based on that kind of structural data, our lab came to this hypothesis that those N-terminal repeat domains may be important for RAB phosphorylation, and it might be that release of what we're calling kind of autoinhibition, where that arm is across the kinase domain, that might be important for RAB phosphorylation. And so, to do this, we are using these tools called DARPins to look at specific conformations of LARC2. So what are DARPins? They're designed to anchor and repeat proteins. As said in the name, they are actually, you can design them for high specificity of your target protein. So you may have this large library of DARPins that are all slightly different in this. Oh no. Oh, here it is. Let me do laser pointer. Yes, in this interaction surface. And so you can have many different DARPins that have high specificity, and you can have a library of DARPins that you can screen for your specific protein and for high specificity. They're very stable and they're inexpensive. You can add fluorescent tags and epitope tags for different microscopy and biochemistry assays. And so we decided to start looking for DARPins that were specific to this RCKW domain, this more interesting catalytic domain of LARC2. And we ended up with two DARPins, E11 and C12, that had high specificity for the LARC2 RCKW. And importantly, with this RCKW protein, they did not change the activity of the kinase. So here you have this inhibitor of LARC2, and you can see a decrease in kinase activity as you increase the concentration of the inhibitor, but you don't see that same effect with DARPins. So for RCKW, it doesn't affect kinase activity inherently. And this work was done in the NAP lab. And so these two DARPins, as I mentioned, they're specific to RCKW, but what we found using some cryo-EM data from Marta in the Lechsteiner lab is that when DARPin C12 binds to the WD40 domain of LARC2, you have this section here where it's kind of hard to see on this because it's also spindly, but it interferes with where that arm actually docks across the kinase domain. And so we would expect for that arm to be much more flexible and not be able to steadily dock across the kinase domain. DARPin E11, on the other hand, although it binds to a very similar position on the WD40 domain, it doesn't actually inhibit or interfere with where that arm is docking at all. And so, of course, the next step was to see, well, do these DARPins affect LARC2 kinase activity in full-length LARC2 kinase activity in cells? And excitingly, if you kind of are following along with me, again, when that arm is not able to dock, you have a more accessible kinase. And so, as you can see from here, you actually have an increase in phospho-Rab over total Rab compared to our DMSO control. So here you have regular kinase activity when there's inhibitor present, and then this increase with DARPin C12. And again, we really think it's due to that DARPin keeping the arm from actually draping across the kinase domain, which makes the kinase more accessible, and Rabs are able to, at a higher rate, come through and be phosphorylated. And so, of course, the next question was, is this true across different mutants of LARC2? And if so, how can we use DARPins to see how those mutations that are not in the kinase domain actually lead to an increase in Rab phosphorylation? Because one of them is over here on a protein-protein interaction domain. How is that actually leading to increased Rab phosphorylation? And so we chose kind of two of the most common mutants in LARC2, this R1441H I'm going to use because that's the version that we had in our lab, and G2019S to look at first. And so again, DARPin C12, it sits on the WD40 domain, lets the kinase become more accessible, and you have this increase in kinase activity, or you have this increase in Rab phosphorylation. With G2019S, which remember is a mutation in the kinase domain itself, you have this baseline increase in kinase activity over wild type, which we would expect. But interestingly, C12 does then further increase Rab phosphorylation. So not only do you have a hyperactive kinase, you have a more accessible kinase. And so you have a further increase in kinase activity. But where it got kind of interesting is that R1441H didn't seem to be affected by DARPin C12. And so, of course, next question is why? But if you remember, I mentioned that DARPin C12, again, when we look at it under cryo-EM, we mostly see the RCKW version of LRRK2, which implies that the arm is undocked and more flexible, and it averages out of cryo-EM. And so you don't see this more stable version of LRRK2 with the arm draped across the kinase domain. You have an accessible kinase. And when Amalia put LRRK2 on a grid and did cryo-electron microscopy, she found that R1441H also had this preference for the more undocked behavior. She saw more undocked LRRK2 on her grids than she saw docked. And so what we think is going on here is that R1441H already has this preference for this more accessible kinase. And so adding DARPin C12 to the mix is not going to then further increase the access of the kinase domain. And so you have kind of a way to induce R1441 like Rab phosphorylation using DARPin C12. DARPin C12 increases Rab phosphorylation in the same manner to R1441H. They both have a preference for an accessible kinase. But going back to DARPin E11, based on cryo-EM data, we saw no real difference in undocked versus docked behavior of that arm. So you saw both this more auto-inhibited, full-length structure on the grid, as well as this undocked RCKW version of LRRK2 on a grid. And so you would think that in cells, it would have no change in Rab phosphorylation. You would have your baseline Rab phosphorylation and then the same amount when DARPin E11 is present. But actually what we see is that Rab phosphorylation of wild type LRRK2 or by wild type LRRK2 actually decreased in the presence of DARPin E11. And so it's not changing any sort of, it's not changing the structural preference of LRRK2. And so the question is, well, why? What's happening? And my current hypothesis is that it's likely due to E11 being more specific and more tightly bound to LRRK2 and actually competing with an activator of LRRK2. So there's potentially some protein that comes in, activates LRRK2 under normal conditions, but when E11 is there, it is no longer able to bind, and so you have this decrease in the accessible kinase. And so to really kind of dig into that and look at it, kind of the next steps in my project are to use DARPin E11 and DARPin C12 to isolate LRRK2 from cell lysates, and then using mass spectrometry and other biochemical assays, see what comes out with LRRK2 when you have either E11 or C12 present compared to our LRRK2 knockout cells. And so to do this is essentially just a co-immunoprecipitation. I've got flag tags on my DARPins, and I mix them with anti-flag magnetic beads. Those are then added to either our A549, which has high levels of endogenous LRRK2 lysate, or LRRK2 knockout lysate, and I then use a magnetic strip to isolate the DARPins, which should isolate LRRK2, and I send it off for mass spectrometry to see what proteins come out with LRRK2. And so far, I've been able to validate this using Western blot and mass spectrometry, but it's easier to show you the Western blot for now. And so as you can see, we've got our regular cell lysate, and when we have, and our input has a little bit of LRRK2. But when we only isolate beads that have no DARPin on them, we don't see anything pulled down with the beads. But when we have E11 present, we're able to pull down some LARC2, and we're able to pull down even more LARC2 with C12. And then we don't have anything come down in our LARC2 knockouts. So proof of concept, the DARPins are able to actually isolate LARC2 from cell lysate. And so the future directions are, of course, to continue with optimizing pull downs and optimizing our mass spectrometry protocols so that we really can look at the differences in what proteins are interacting with LARC2 in different conformations, as well as to test these other LARC2 mutants. So I didn't really touch on them, but there's this mutation in the core B domain that is very prevalent in different populations, this Y1699C. There's another mutation in the kinase domain that's also prevalent, I2020T. And then I think probably one of the most interesting ones that we'll get to look at is this G2385R, which is a mutation in the WD40 domain, not terribly far from where my DARPins bind. And so I think that one could be really cool just in that it's, again, another structural change that happens to LARC2 when you have this mutation in a domain that has nothing to do with any catalytic activity whatsoever. And finally, to kind of look at the cellular effects of what happens when you have that arm open behavior. So does it change where LARC2 is localized in the cell, or what substrates it prefers, and those sorts of things. So I also have a lot of microscopy in my future. And that's really it for me, you guys. Again, want to thank Sam and Elizabeth. It's been a really fun project that I've really enjoyed so far. Yeah, that's it.
We do have a few minutes. Does anybody have questions for either of these presenters? No? Well, don't go running. Oh, I'm sorry. Go ahead.
Why did you choose DARPins as your binders as opposed to antibodies or SCFPs?
That's very good
I'm
question.
sorry, would you repeat the question, please?
Oh, yes.
For the back.
Yeah, so the question is why did I choose to use DARPins instead of antibodies? And honestly, the LARC2 antibodies thus far have just not been very good. They're not specific. They're okay in a Western blot, but you have to like completely separate the top of the blot where LARC2 is from the rest of the blot because it'll just bind everywhere. In IF, you just can't really tell. There's so much background, you can't tell what's actually LARC2 and what's not. And for immunoprecipitation, it's just been completely non-specific. So that was kind of the main reason. This project kind of originated as a way to provide the community that works with LARC2 with new tools. And I think so far it's insane the amount of LARC2 it actually pulls out on mass spec. Like we've been using some like inhibitors, like tagged inhibitors of LARC2 to also pull it out from cell lysate. And it does well, but the DARPins are just really tight binding. They're really good tools. Yeah.
Somebody else? For Jillian or or Catherine? Okay. Thank you both. Appreciate it. Good job.